Welcome to the video on the bacterial cell wall. This is one of the most important structures of the prokaryotic cell wall because it helps maintain the cell shape, protects it from physical damage, and prevents the cell from bursting when water moves into it, which we talked about with passive transport and osmosis. Because bacterial cell walls differ in their structure, we also use these differences to help categorize bacteria into one of two categories, either gram-positive or gram-negative. So, regardless though of which category they belong to, the cell wall is primarily made up of peptidoglycan. And peptidoglycan, if you look in the picture below, consists of repeating units of NAG and NAM. Now, you don't need to know what those stand for because, well, it's a mouthful. But if you look closely, you'll see that it goes a blue NAG and then an orange NAM and then a blue NAG and an orange NAM. And so that's going to be the basis of the peptidoglycan. One of the big differences between gram positive and gram negative is how much peptidoglycan they have. Peptidoglycan is very thick with gram-positive bacteria. And because of that, because of those many layers of peptidoglycan, you'll notice in this bottom left picture that the NAM molecules have these four little amino acids coming down, and then it attaches to a neighboring NAM using a five amino acid connecting chain. So the four amino acid chain is called a tetrapeptide. Again, that's what you're seeing here in green. And then there's the pentapeptides that are connecting those neighboring chains of NAG and NAM. And so this forms this very complex, thick layer of peptidoglycan with our gram positives. Our gram negatives have a very thin layer of peptidoglycan, sometimes just one or two layers. So you'll notice that there's the repeating structures of NAG and NAM. Again, you'll see that NAM has the four tetrapeptides coming down off of it, but there's no pentapeptide that's connecting between them, like you see in the gram positives. So the amount of peptidoglycan is just one difference between the gram-positive and gram-negative bacteria. So let's go ahead and zoom in and take a closer look at both of these structures. So as I mentioned, both the gram-positive and the gram-negative bacteria contain peptidoglycan, but they organize it very differently. So on the left, excuse me, is going to be a picture of the gram-positive cell wall. And you'll notice that the gram-positive has this thick layer right here of peptidoglycan surrounding that plasma membrane. Okay, so take a look at this picture, and you'll notice right here, let me get the highlighter. Here is our plasma membrane. Okay, so we're focusing right here on this left-hand image. We've got our plasma membrane, and then we have this big, thick layer of peptidoglycan. Okay, but now if you check out the right-hand picture, you'll see again the plasma membrane here, but then there's a very thin layer of peptidoglycan right there. Okay, and that can actually be problematic because of the role of the cell wall in maintaining structure and preventing the cell from lysing should water rush in. And so essentially the cell has to kind of compensate for that. And so you'll notice right here outside of the thin layer peptidoglycan is a second membrane. Okay, it's another phospholipid bilayer, and we refer to it as the outer membrane. So the outer membrane is really, really important because it's providing these gram-negative bacteria with this extra layer of kind of protection, an extra protective barrier. It contains two important molecules. One would be porins, and these are little pores that act as selective channels that control what can and cannot pass through the membrane. These porins have a very specific structure, so only molecules with a compatible or complementary structure can pass through it. Then also part of the outer membrane is a molecule called lipopolysaccharide. We refer to it as LPS for short. And this is actually an endotoxin. And so these two components of the outer membrane really have some interesting effects on people during an infection. So these porins here can make it really difficult for antibiotics to be effective because the antibiotics have to be able to cross this outer membrane to even try to gain access to the cell. And then this lipopolysaccharide, like I mentioned, acts as an endotoxin. And so should this bacteria lyse or be destroyed, these little endotoxins are going to be released inside the body, which can have some negative effects. So notice how complex this gram-negative cell wall is. So again, it's going to have a thin layer of peptidoglycan, and then it's going to have an outer membrane, which is another phospholipid bilayer. But associated with the outer membrane are those selective channels called porins and a molecule called lipopolysaccharide. Okay. And again, just to go back and compare the gram positive, we've got a really thick layer of peptidoglycan. And it also has a molecule called lipoteichoic acid that you can see here in green that's kind of anchoring here to the plasma membrane and kind of helps maintain this structure of this thick layer of peptidoglycan. All right, so we're going to wrap up this video with a quick explain it. So a patient has a gram-negative bacterial infection and is prescribed an antibiotic. Explain why gram-negative bacteria are often more difficult to treat and why the patient may feel worse before actually getting better. So at the beginning of treatment, they would feel worse. Use the structures of the gram-negative cell wall in your explanation. So with this, we need to go back and think about the difference between a gram-negative and a gram-positive bacteria. What is it about the gram-negative bacteria that can explain for these issues, the difficulty in treating with antibiotics and possibly feeling worse before feeling better? So remember that both gram-positive and gram-negatives have some peptidoglycan, but our gram-negatives have the thin layer, and so they also have that outer membrane to help compensate for that. And so there are two important pieces of that outer membrane, and one of them was the porin. So remember that porins have a very specific shape, and only molecules that can fit through that porin can gain access to the bacteria. So this is why gram negatives are often more difficult to treat, is because the presence of those porins. And then the other big component of the outer membrane was LPS, or lipopolysaccharide. And the lipid portion of LPS is referred to as lipid A, and that acts as an endotoxin. So as gram-negative bacteria are damaged or killed, LPS can be released. This can trigger a strong inflammatory response, and it can actually result in things like fever and inflammation. And so sometimes by killing the bacteria, it can actually intensify the patient's symptoms before the infection begins to improve. So it all comes down to the structure of the gram-negative cell wall. All right, y'all, that's it for this video. Please let me know if you have any questions.